All right, welcome back. So we're looking here at the structural and contractile unit of the skeletal muscle. It's the sarcomere. The sarcomere, as we mentioned, it's the region between two successive Z discs, as is shown on this diagram on the side. It's composed of alternating thick and thin filaments. They are made of contractile protein. As you remember, the thick filaments are formed by the protein subunits called the myosin, and the thin filaments are formed by the protein subunits actin, troponin, and tropomyosin. Thick filaments will run through the entire length of the A band or the dark band. If you remember, this is the dark part at the center of my sarcomere. Thin filaments will run the entire length of the I band and part way of the A band. So the part where I have the overlap between the thick and thin filaments, it's gonna be also part of the A band or the dark band. Z discs, those are cone-shaped sheets of proteins that anchor the thin filaments and connect the myofibrils to one another. If you remember on here, those are my Z discs that are connecting the thin filaments to one another. The H zone is this central zone where I don't have overlap between the thick filaments and the thin filaments. It consists only of thick filaments. The M line is the line of protein that holds the thick filaments to one another, running at the center of the sarcomere. Look it on here. This is my M line attaching the thick filaments to one another. The A band, this is a part that consists only of, that will appear darker in color as under the microscope, is a striation. It consists of mainly the myosin and partly the actin and the troponin and tropomyosin of the thin filaments. On the other hand, the I bands, those are the light colored parts that consist only of the thin filaments. For our first in-class activity for today, the Z-disc hold what together? You remember what is held together by the Z-discs? Are you holding the thick filaments, sarcomeres, the I band, thin filaments? Z-discs are attaching what together? I see here many people are saying sarcomeres. What is held in here by those Z discs together are not the sarcomeres, but what in here? My thin filaments. Thin filaments. Thin filaments. Those are what is held by the Z discs. They are just at the connection between the successive sarcomeres, but actually they are holding together the thin filaments. All right, so the good answer here was going to be D. Thick filaments are composed of protein subunits called the myosin. The myosin heads form the cross bridges with the actin of the thin filaments, and they have binding sites for ATP. Remember, the muscle contraction requires a lot of energy for it to take place. So in order for me to form the cross bridge, I need to hydrolyze ATP. I need to break down ATP to release the energy needed for me to form this cross bridge. Thick filament is going to be composed of proteins, actin, tropomyosin, and troponin. Actin has the myosin binding sites, if you remember. The binding sites on the actin are going to be exposed in order for the muscle contraction to take place. So we've got the binding sites on the actin subunits, where the myosin heads are going to be forming the cross bridge. Looking again here, this is the myosin, and this is the head of the myosin that will form the cross bridge. If you notice on here, it has a binding site for the ATP. This is where the ATP will come and bind and become hydrolyzed to create the energy needed for the cross bridge to be formed. Looking on here, you can see the actin subunits, and on each of the actin subunits, you've got the myosin binding site. In a relaxed state of the muscle, those binding sites, as you see, are covered with the tropomyosin. In a relaxed state of the muscle, you're covering those binding sites on the actin subunits by the tropomyosin. Remember, inside your skeletal muscles, you've got the sarcoplasmic reticulum, and this is a smooth endoplasmic reticulum that will be surrounded by the myofibrils, the thick and thin filaments. The function here of the sarcoplasmic reticulum will be to regulate the calcium levels in the cytoplasm. If I've got a signal, the sarcoplasmic reticulum gonna release calcium out. If I don't have a signal, so the muscle shouldn't be contracting, so it will be collecting this calcium back from the cytoplasm to the inside of the tubules. And this what creates the different cytoplasmic levels of calcium. And according to the calcium levels in the cytoplasm, the muscle will either contract or relaxes. Remember, what are the T tubules? Those are continuous with the sarcolemma. They penetrate the cell at the junction between the A and I bands. So those are the invaginations of the sarcoplasm of the sarcolemma, allowing you to be connected to the inside of the cell. See on here, those are my T tubules, and they are connected to those tubules of the sarcoplasmic reticulum. Remember, whenever I get a signal down to the T tubules, this will cause the excitation of the endoplasmic reticulum, causing the endoplasmic reticulum to release the calcium in the cytoplasm. Sliding filament model. In a relaxed state, the thick and thin filaments overlap slightly. During the contraction, the myosin heads bind to the actin, detach, bind again, propelling the thin filaments to slide towards the M line. So I'm putting here those thin filaments towards the M line. So as I contract, sliding those thin filaments along the thick filaments, the H zone shortens and eventually disappears. This part where I don't have overlap between the thick and thin filaments will disappear as I contract, because simply all the fibers gonna be overlapping one another, and I won't have this area where I have only thick filaments and not thin filaments overlapping with it. What this results in? It results in a shortening in the length of the sarcomere, and as a result, you're gonna have a shortening of the length of the whole muscle. This is in a relaxed state. I see clearly the M line on here, the H zone, and this is my A band, and this part on here is my I band. So as I pull the thin filaments towards the M line, the H zone disappears, so no H zone on here. And the two Z discs attaching the thin filaments to one another are gonna be moving closer towards the M line, causing the shortening in the length of the sarcomere. What happens at the neuromuscular junction? Skeletal muscle cells are stimulated by the somatic motor neurons. The axon terminal and the muscle fibers are separated by a gel-filled space called the synaptic cleft. So if we're looking here at the junction between the nerve cell and the muscle cell, this space is filled with a gel-like substance. We call this is the synaptic cleft, the junction between the terminal end of the nerve cell and the muscle cell membrane. Synaptic vesicles of the axon terminals containing the neurotransmitter acetylcholine will be releasing their contents in the synaptic cleft if I received a signal down from the brain. So again, what we're looking at in here is the junction between the nerve cell and the muscle fiber. What happens again? Once I receive a signal, this will cause the opening of calcium channels, because those are my voltage-gated calcium channels. The influx of the calcium gonna stimulate the vesicles containing the neurotransmitters to fuse with the cell membrane of the nerve cell, releasing the neurotransmitters in the gel-filled space between the nerve cell and the muscle cell, because this is my synaptic cleft. As I release the neurotransmitter in the synaptic cleft, the neurotransmitter here, acetylcholine, binds to its receptor, causing the opening of channels, which will allow the sodium in. This sodium, as it travels inside the cell membrane, this will cause the change in the membrane polarity. So inside of the cell will become more positively charged as you allowed the sodium in. Compared to the outside, is going to be more negatively charged. This electrical signal is going to keep traveling along my sarcolemma to reach the T tubules. Once it travels to the T tubules, it causes the opening of the calcium channels, enhancing the endoplasmic reticulum or the sarcoplasmic reticulum to release lots of calcium out in the cytoplasm, as you see on here. So once I receive the signal down to the T tubules, the sarcoplasmic reticulum release the calcium out in the cytoplasm. Calcium out in the cytoplasm, the calcium will bind to the troponin, causing a conformational change of the troponin. Means what? Means it changes the shape of the troponin. And as I change the shape of the troponin, I will be pulling the tropomyosin away from the binding sites on the actin subunits. So you see on here, this is in a relaxed state. The tropomyosin is covering the binding sites on the actin subunits, but when I get the calcium out of the cytoplasm, calcium binds to the troponin, moving away the tropomyosin, exposing the binding sites on the actin subunits. What's going to happen next? The myosin will form a cross bridge. So the heads of the myosin subunits of the thick filaments will form a cross bridge and will be pulling the thin filaments towards the M line, causing the shortening of the length of the sarcomere, allowing the contraction to take place. At low intracellular calcium concentration, the tropomyosin is blocking the active binding sites on the actin subunits. The myosin heads cannot attach the actin, and the muscle fiber is going to be relaxed. This is what we've seen on here. Tropomyosin is covering the binding sites on the actin subunits. Myosin heads are not able to attach the actin subunits. What happens if I have high intracellular calcium levels? Once I get the signal, in sarcoplasmic reticulum going to be releasing the calcium out in the cytoplasm. This is going to increase the calcium levels inside the cytoplasm. Calcium binds to the troponin. The troponin changes its shape, moving away the tropomyosin from the binding sites on the actin subunits. And as I expose the binding sites on the actin subunits, the myosin heads will form cross bridges, binding, bending, detaching, and binding to the next one, forming what we call the cross bridge cycle. This is how the cross bridge cycle is gonna look like. So you start first by binding, and in order for me to bind, the head of the myosin needs energy. So it's gonna be breaking down in ATP. As I bind, I will bend, pulling the actin towards the M line, causing the sliding of the thin filament. Remember, in order for you to detach, to unbind from the actin, this requires you to have an ATP. So you see, for example, in a game, somebody is running so fast and all of a sudden he got a cramp. He's not able to keep contracting and relaxing. So his muscle is stuck in a contracted state. Why? Because in order for you to relax, remember, you need energy. So if this person did consume all the energy, he doesn't have any more energy in his muscle cells, the muscles will be kept contracted and he fails to relax the muscle. So in order for you to unbind the myosin heads from the binding sites on the actin subunits, you're gonna need to bind to ATP. I need energy to bind, I need energy to unbind as well. You bind to the ATP, you will unbind from the binding sites on the actin subunit, and you're ready to bind to the next one. You bind to the next actin, you bend, pulling the actin, allows the sliding of the thin filament. You unbind. In order for you to unbind, you need the ATP to bind to the myosin head, and you're ready to bind to another actin. You bend, pulling the thin filament. As you bend, you're sliding the thin filament along the thick filament, and then you unbind to bind to the next one. You bind, bend, detach, bind, bend, detach, bind, bend, detach. A cycle here is what we call the cross bridge cycle. This will take place in order for you to allow the sliding of the thin filaments along the thick filaments. If we look at here at the very basic muscle contraction, you can see that the muscle contraction starts by a latent period. I've received the stimulus. I have a latent period. And after this latent period, you're going to see an increase in the muscle tension. This is where I have the contraction taking place. I'm reducing the length. I'm increasing the tension of the muscle. This is where, when the contraction is taking place, because this is a contraction phase of the muscle twitch. This is a basic form of a muscle contraction. And then what happens after you reach the peak of the tension that you want? You're gonna keep relaxing. So third phase of a muscle twitch, it's the period of relaxation. So 3 phases on here. I have the latent period. This is the time needed for the excitation contraction coupling. So what happens during this latent period? In those few milliseconds, all what we have discussed. The acetylcholine binds to the receptor, allowing the sodium in, creating a depolarization of the cell membrane. This electrical signal travels down to the T tubules, causing the opening of calcium channel, stimulating the endoplasmic reticulum to release the calcium in the cytoplasm. The calcium binds to the troponin, changing the shape of the troponin to move the tropomyosin away from the binding sites of the actin subunits, and the myosin heads will form a cross bridge to bind to the actin, pulling the actin, starting the contraction. So all what I just mentioned is taking place in those few milliseconds. This is what we call the excitation contraction coupling. The time between the increase in the tension, the shortening of the length of the muscle, and the receival of the signal. This is called my latent period. After the latent period, you start the shortening of the length of the muscle fibers, and this will increase the tension of the muscle fibers. This is what we call the contraction period, in which I have a high intracellular calcium level. During the relaxation phase, I'm putting the calcium back in. So this calcium will not remain outside like this, binding to the troponin. I need to reuptake this calcium back to the sarcoplasmic reticulum. Lowering the calcium levels in the cytoplasm gonna allow you to relax your muscle. And the tension will gradually decline all the way to 0. So again, what is the basic muscle contractions we're looking at in here? It's called the muscle twitch. Muscle twitch is the minimal contractions that you can see on here. You start by a latent period. This is when the excitation contraction coupling is taking place. You increase the tension within the muscle. Increasing the tension, this is why period of contraction, during which you are releasing the calcium out in the cytoplasm. The third phase is my relaxation phase, when I will be reuptaking this calcium to the inside of the sarcoplasmic reticulum. Decrease of the intracellular calcium level going to result in the relaxation of my muscle cells. So what will affect the strength of the muscle contraction? Would you be performing the same contraction, moving a pen, for example, and moving a table? What do you think? Do you create the same degree of contraction? Do you exert the same force in both cases? What do you think? So is your muscle either fully contract or fully relax, or you have different grades of contraction? What do you think? All or none, or grades of contraction? I have grades, different grades, definitely. So if I'm moving a pencil or a pen, for example, what's going to happen? I don't need to enhance all the muscle fibers within my biceps to contract, to perform a maximum contraction. Compared to if I'm holding something really heavy, I will need much more fibers to contract. So you have in your skeletal muscles what we call a graded muscle contraction. According to what, you're going to be changing the grade of the contraction in response to the frequency of stimulation. How many stimuli in a period of time did you receive? So frequency is number in time. Excuse me. And the other factors that affect the grade of muscle contraction will be the strength of the stimulus. How strong is the stimulus I am receiving to the muscle? So two main factors here that will affect the grade of the muscle contraction. First one is the frequency of stimulation, how fast the stimulation is taking place to the muscle. And the second factor is going to be the strength of the stimulus. All right, so let's look on here. I've got one stimulus, a threshold stimulus. What I mean by a threshold stimulus, this is a minimal stimulus that allows a minimal muscle contraction, a noticeable muscle contraction. This is what we call a threshold stimulus. Threshold stimulus. So I've got a stimulus, a single stimulus. I have a latent phase, contraction phase, and relaxation phase. That's it. What if I got another stimulus before I complete the relaxation? Before I reduce the tension all the way down to zero. This means what? Before complete relaxation means I didn't reuptake all the calcium back. So I still have some of the calcium outside in the cytoplasm. So let's say if I receive the stimulus, I would be releasing 4 calcium out to bind to 4 troponin, to move 4 tropomyosin to bind, to allow 4 myosin to bind to act. All right. In order to fully relax, what I need to do, I need to reuptake those 4 calcium back to the sarcoplasmic reticulum. If I didn't allow a complete relaxation and I received another stimulus, so the second stimulus is going to result in the release of another 4 calcium. So during the partial relaxation, I did reuptake 2 calcium, but I still have in the cytoplasm in here, I still have 2 calcium. So how many calcium are still remaining if I didn't perform a complete relaxation? I did reuptake only 2 from the previous stimulus, and now I released 4 calcium. So how many do I have in total outside in the cytoplasm right now? I have 6 calcium. 6 calcium means what? Means I will be binding to 6 troponin, moving away 6 tropomyosin. So I will have more exposure, getting a stronger contraction. So you will notice that as I didn't complete the relaxation, I got only a partial relaxation, the next contraction is going to be of greater tension. It's going to be stronger. I didn't allow a complete relaxation. So let's say I did reuptake 3 or 2. I did reuptake 2 calcium, and I've got a third stimulus. And third stimulus caused the release of 4 calcium. So I did have 6 calcium. I did reuptake 2. So how many are still remaining from outside in my cytoplasm? I still have 4. And the third stimulus was received, and this caused the release of 4 calcium. So how many calcium do I have right now out in the cytoplasm? I have a total of 8 calcium. So the previous time I did have 6. The previous time I did have 4. So 8 calcium means what? Means I will bind to 8 troponin, moving away 8 tropomyosin, having more exposure to the actin subunits, stronger contraction. So the next contraction, as I don't allow a full relaxation to take place, is gonna create a much greater tension in the muscle. Why this is happening again? If you don't allow a complete relaxation, you're gonna still have calcium from the previous contraction outside of the cytoplasm, as you didn't reuptake all the calcium to the inside of the endoplasmic reticulum. This is gonna cause an increase in the force of contraction. So as I increase the frequency of stimulation, faster stimuli prevents the complete relaxation of the muscle. As I increase the frequency of stimulation, the contraction gonna be stronger. What if you received a very, very fast stimulation, a very high frequency of stimulation, very high frequency of stimulation? What's gonna happen? You're gonna cause the excitation of the sarcoplasmic reticulum, and this is gonna result in a release of huge amounts of calcium out in the cytoplasm, binding to all the troponin, moving away all the tropomyosin, and you're gonna reach the maximal possible tension in the muscle. And this gonna be contracted as long as you receive the stimuli, causing you to create a continuous contraction of the muscle, or tetanic contraction, or tetanus. Remember, like your back muscles. Now, while you're sitting down or standing up, your back muscles are performing tetanic contraction, continuous contraction. What is the concept on here? The brain is sending very, very high frequency of stimulation, keeping the muscle in a contracted state, continuous contraction, not allowing any relaxation to take place. All right, again, the higher the frequency of stimulation, the stronger the muscle contraction, as you are not allowing a complete relaxation to take place. So the amount of calcium available outside in the cytoplasm is gonna be higher every time you receive a stimulus. Another factor that affects the strength of the muscle contraction is going to be the strength of the stimulus it receives. The minimal stimulus that you're going to be responding to create a muscle contraction, it's what we call the threshold stimulus. So if this is the voltage of the stimulus, I received a stimulus lower than the threshold stimulus. Would there be any contraction taking place in my muscle? What do you think? Would I have any increase in the tension? Would I have any contraction? Yes or no? Again, threshold stimulus is the minimal stimulus that you will respond to. If you've got a stimulus lower than threshold, would you have any contraction? No contraction. No contraction. Why? Because simply it was not enough to cause the excitation of any muscle fibers. I didn't have enough strength to recruit any of the muscle fibers in this muscle. How about if I have a stronger stimulus than the previous one, but still lower than this threshold stimulus? Would I see any muscle contraction? What do you think? If it's still lower than the threshold, no muscle contraction will be taking place. What if I've got a threshold stimulus? Threshold stimulus is a minimal stimulation. Would I have any muscle contraction? Yes, you're going to have a minimal contraction on here. Why? Because some of the muscle fibers got recruited. Minimal contraction, minimal contraction. As you keep increasing the strength of the stimulus beyond the threshold, so the minimum is the threshold, I need to get higher stimulus than the threshold. As I keep going up, increasing the strength of the stimulus, more muscle fibers gonna be recruited, and as more muscle fibers gonna be recruited, I will be creating a stronger muscle contraction. Until when? Until I receive a maximal stimulus. Once you receive a maximal stimulus, all the muscle fibers in your muscle gonna be recruited. All the muscle fibers gonna be recruited. And this will allow you to reach the maximal tension by the muscles. This is the maximal contraction that you can create. What if you've given me a threshold, a stimulus greater than the maximal stimulus? Would you have any contraction stronger than the maximal contraction? Yes or no? What do you think? No. Why? Because all the muscle fibers in the muscle are already stimulated. All of them have been recruited. So I can't perform any stronger contractions than the maximal contraction. So if, for example, we have a wall in here and we are 20 people in class, I started to push this wall on my own. So I'm creating this amount of tension. I told you, all right, so some people, I would like some people to help me pushing this wall. So 5 of you decided to help me. So the tensions that we are applying now on the wall is gonna be greater. So in order to convince more people of you to push this wall with me, I told you I would be giving you 5 extra credit points if you're gonna be pushing this wall with me. So more people got convinced, and I've got 10 more people pushing this wall with me. So the amount of tensions that we're applying on the wall is gonna be higher. So I told you, all right, so rather than 5 extra credit points, I will give you 10 extra credit points for those who will be pushing this wall with me. So all of you got convinced, and the remaining 5 people that were not pushing the wall, when I told you that you're going to be receiving 10 extra credit points, this is the stimulus on here, more people got recruited, and you all started to push the wall. So all of the 20 people in the class right now are pushing the wall, and this is the amount of tension we're applying on the wall. If I told you you're gonna get 50 extra credit points, I'm increasing the stimulus on here. Can we increase the amount of tension applied on this wall? No. Why? Because all of you are already have been recruited. This is the maximum number of the people in class. All the 20 are already pushing this wall. So I can't create any further tension on this wall. Even if I go to 1,000 extra credit points, there will be no increase in the amount of tension applied to the wall because all of you have been already recruited, just giving you 10 extra credit points. So what do we call this 10 extra credit points that will recruit all of you to push the wall? It's the maximal stimulus. And the tension that all the 20 of you gonna be applying on the wall, it's gonna be the maximal contraction. If I increase the stimulus, I go 50 extra credit point, 200 extra credit point, would there be any increase in the degree of tensions that we can ever create on this wall? No. Why? Because simply all of us are already pushing the wall. All right, so Theodore here got stimulated at 5 extra credit points. All right. All right, questions. So we've got two main factors that will affect the force of contraction or the degree of tension created in the muscle. First one was the frequency of stimulation. Remember, the higher the frequency of stimulation, the less likely you're going to be relaxing in between the contractions, and so you're going to have higher levels of tension created because less calcium going to be reuptaken. You then allow full relaxation to take place. But here, the strength of the stimulus doesn't have anything to do with the calcium. It has a role to play with the number of fibers that you're going to be recruiting in the muscle. The higher the strength of the stimulus, the more muscle fibers, the more cells that will get excited. So here, if this is my muscle, I have, let's say, 1,000 muscle fibers. The stronger the stimulus, the more muscle fibers out of this 1,000 going to be recruited to perform the contraction. All right, is this clear, Alexandra? So in a leg cramp, leg cramp might be due to many reasons. So the one example that we've mentioned, if somebody was running so fast and all of a sudden he got a muscle cramp that doesn't allow him to relax the muscle, remember, this is due to the lack of energy, because in order for me to relax, to detach the myosin heads from the actin binding site, I need energy. So if I already did consume all the energy while I'm contracting, I don't have any further energy, ATP present in my cells, so I will fail to detach the myosin head from the myosin binding site of the actin subunit. So this is what causes the cramp. Also, iron levels disturbance, if a person doesn't have enough potassium in his cells, can cause cramps. That's why you'll see people going to the gym with a banana. Bananas are rich in potassium. So many disturbance in the ionic levels can result in disturbance in the electrical conduction and normal contraction, relaxation of the muscle fibers. All right, but the examples that we've mentioned concerning the muscle cramp is going to be due to lack of energy, and this is going to cause the failure of the muscle relaxation. All right. Another factor that affects the force of contraction is going to be the size of the muscle fiber. We have three sizes of the muscle fibers. Small muscle fibers will be performing this amount of tension. A medium muscle fiber definitely going to perform a stronger contraction compared to the small ones. And the large muscle fibers will be performing a greater tension compared to both the medium-sized muscle fibers and the small-sized muscle fibers. So the larger the muscle fiber that you are activating, the larger the muscle cells that you're activating, the stronger contraction it can perform. That's why if you train your muscle, you're not increasing the number of muscle fibers, you're not increasing the number of muscle cells, but you are increasing the size of the muscle fibers. So like bodybuilders, they have larger muscle fibers, not more muscle fibers. And again, what we mean by muscle fibers are the muscle cells. All right, so here I'm not increasing the number of muscle fibers, I am increasing the size of the muscle fiber. So the larger the size of the fiber, the stronger contraction it can perform. So a bodybuilder can hold much higher weights because he has larger muscle fibers. All right, does this make sense? All right, so if you are fully relaxing on the beach, is there any muscle contraction in your muscles? You are fully relaxed, you're not moving. Is there any muscle contraction in your muscle? Actually, always there will be a slight muscle contraction. All your muscles, even when you are fully relaxed, when you are sleeping, there will always be a slight contraction in your muscles. We call this is the muscle tone. Why do we have a muscle tone in our muscles? For our muscles to stay firm, healthy, and ready to respond. So rather than every time you decide to contract the muscle, you start from scratch. You don't have any contraction in the muscle, and you're gonna need to build up a higher tension. You're gonna keep your muscles at this level of tension for it to be easier and faster for you to perform the required contraction. So when you are fully relaxing in a camp, for example, if you're camping, you still have some degree of contraction in your muscles. We call this slight degree of contraction. It's my muscle tone. Why do we need this? Remember, in a camp, while you are relaxing, you might have an alligator getting closer to you all of a sudden. So what would you like to do? You are ready to run. So rather than starting from a zero tension, you're gonna start from a much higher tension, which is the muscle tone. Makes you ready to respond, faster response of the muscle contraction. All right, does this make sense? Does this make sense? All right, so one third question for today that we skipped on here. So what neurotransmitter is it required for muscle contraction? What is the neurotransmitter that we've released from the nerve cell to bind to the muscle cell in order to initiate the muscle contraction. Can you remind me, what do we call this neurotransmitter here? Don't remember, what is the neurotransmitter here? Acetylcholine, exactly. Acetylcholine. I believe this is the third, yeah, maybe, but let me double check. Yeah, it seems this is the second question. So this is the second question. Second question. This is question number 2. All right. This is question number 2 of the in-class activity for today. All right, so one other assignment for today that you need to complete as part of the in-class activity for today is to write down what you understand from this video from YouTube. All right, this is going to be your third question or assignment. It's going to be an essay question. It's going to be an essay question. It shows you in a video animation how to perform the muscle contraction. So look up here on your YouTube. I'm sending out the link in the chatting box right now. Look up on YouTube, muscle contraction process HD animation. Muscle contraction process HD animation. Please watch this video and write down what you've understand from the video, which is simply what we have been discussing today in our meeting. So it's a brief video that explains in a very good animation format how the muscle contraction takes place. So this is going to be the third question or the third item of the in-class activity for today's lecture. All right, so again, please watch this video recording and write down an essay summarizing what you understand from this video recording. It's going to be a paragraph here. Yeah, like I don't put any limit for the number of words, but it needs to deliver the whole concept that has been discussed with its details. All right, so you need to write down what you've understand from the video recording. All right, there is no minimum or maximum number of words that is required, but you need to deliver the idea of the video and the details discussed in the video recording. So it can't be 4 or 5 lines. It needs to be at least 15 to 20 sentences at least. All right, any questions? Any questions? Just